Dear student, the topic of this video is the physiology of pregnancy. Pregnancy can be divided into 4 stages. First is the initiation of pregnancy, which includes the different stages like fertilization process, then the implantation of the zygote or the blastocyst, then different hormonal secretion, how the pregnancy is maintained, followed by the period of gestation when the fetal will grow. After that, there will be parturition, that is a process of labor, and followed by the lactation or the milk production, secretion, and ejection. So in human, fertilization of the ovum by the sperm, it usually occurs at the ampulla of the uterine tube or the fallopian tube. So you can see the areas of the fallopian tube. So this area is your, this is the fimbria part from where the ovum will be, after ovulation, the secondary oocyte will be taken up, and this area is the ampulla, this is the isthmus, that is the junction between the fallopian tube and the uterus. Okay, so at the ampulla actually there will be fertilization. The day of the fertilization will be taken as a day 0. Okay, so how actually this fertilization occurs, that we will see now. So we know that millions of sperm are deposited in the vagina after copulation, and eventually what happens, out of this million sperm, 50 to 100 sperm, they will reach the ovum, and out of this 50 to 100 sperm, few of them, they will come in contact with the surrounding covering of the ovum. Okay, now what are the coverings or surrounding barriers of the ovum? So ovum, you can see here, so this is the actual ovum, this one, this is our ovum here. Okay, and so this is the plasma membrane of the ovum. Outside the plasma membrane of the ovum, this membrane is known as a vitelline membrane, and this particular space is your perivitelline space, that is the space between the plasma membrane and the vitelline membrane. Now outside this vitelline membrane there is a thick gel coat. This area is a thick gel coat, you can see. This is your zona pellucida. This is your zona pellucida. Okay. So ovum surrounded by its nuclear, its membrane. After outside of this membrane there lies the vitelline membrane. In between this there is perivitelline space. Okay, and outside this vitelline membrane, this jelly layer is your zona pellucida. Okay, outside the zona pellucida, what is this layer? This is your corona radiata. Okay, so these are the layers that the sperm has to penetrate in order to reach the ovum. Okay, so what are the steps by which it will penetrate these layers? So first it will penetrate the corona radiata. So corona radiata, it is an intracellular matrix consisting protein and which contains a high concentration of hyaluronic acid. So this hyaluronic acid will be hydrolyzed by the release of hyaluronidase that will release from the sperm head. So here in this picture we can say there is no corona radiata. That is, it is totally hydrolyzed by the hyaluronidase enzyme which is released by the sperm. Now the sperm, it is in contact. So now the dispersal of corona radiata, after dispersal of corona radiata, the sperm comes in contact with the zona pellucida. So sperm contacts with the zona pellucida. The step number 2 is when the sperm will come in contact with the zona pellucida, there will be acrosomal reaction. So there will be release of acrosomal content which contains so many hydrolytic enzymes which will help the sperm to penetrate this gel coat of zona pellucida. Okay, so when after dispersion of corona radiata, then sperm will come in contact with the zona pellucida. Then there will be acrosomal reaction and zona pellucida will be penetrated, followed by the acrosome reacts with the perivitelline space. I told you that this is a vitelline membrane and this is the plasma membrane of the ovum. So the space in between is your perivitelline space. So now the acrosome reacts with the perivitelline space. That is the step number 3. Now the step number 4 is plasma membrane of the sperm and the plasma membrane of the ovum. They will fuse together. So there is a fusion like this. Okay, so when the fusion is done, the sperm nucleus will enter within the ovum. Okay, and after that, in order to prevent, when one sperm has entered within the ovum, the prevention of other sperm to enter into the ovum, it has to be prevented. Okay, in order to prevent polyspermia, so the cortical granules will fuse with the egg plasma membrane. They will form an impermeable barrier. Okay, so these are the steps. The first is the grossly, these are the steps, that is corona radiata will be dispersed, then it will come to the in contact with zona pellucida, then there will be acrosomal reaction which will facilitate to penetrate the zona pellucida, and then acrosome reacts with the perivitelline space. After that, the plasma membrane of sperm and the egg will fuse, entry of sperm nucleus within the ovum, and prevention of entry of other sperm into the ovum. So these are the steps in gross. Now we want to see the molecular detail of this each of these steps. So this is the corona radiata. It has been penetrated by the hyaluronidase and the sperm has come in contact with this jelly coat of zona pellucida. So what will happen now? So before going to that, we must come to our brush up, a quick brush up of the structure of the sperm. Sperm has a head. This one is a head. It has a middle piece. It has a tail and the end piece. Okay, and in the head you can find the nucleus here, and there is an acrosomal vesicle. Okay, so this is the acrosomal vesicle, and the whole thing, it is surrounded by the plasma membrane of the sperm. Okay, now this acrosome is a lysosomal like compartment that is derived from this acrosome. This acrosome is a plasma membrane like thing, that is a lysosomal like thing, that contains huge amount of different type of enzymes. Okay, so it has a very low pH, contains soluble hydrolases. Soluble hydrolases, these are the serine protease acrosine. They contain what? They contain acrosine. Acrosine. This acrosomal vesicle, they contain the serine protease acrosine. Okay, so during acrosomal reaction, what happens? You can see that this acrosomal vesicle has an outer membrane and an inner membrane, and outside them there is this yellow plasma membrane. We can see that. So what happens during acrosomal reaction? There will be fusion between the outer layer of the acrosome with the plasma membrane, and through this fusion there will be excretion of the enzymes. There will be exocytosis of the enzyme. Okay. So how this acrosomal reaction will be initiated? That will be initiated by the combination between the sperm head and the zona pellucida. So you'll see that the zona pellucida, it is a 13 micrometer thick in human. So zona pellucida, it is 13 micrometer thick, and it has 3 sulfated glycoproteins. What are those 3? One is a ZP1, zona pellucida 1, zona pellucida 2, and the zona pellucida 3. So we can see that this yellow one is the zona pellucida 1, and this orange, this is ZP3, and this blue is our ZP2. Now what happens, this ZP2 and ZP3, they combine to form the basic unit that polymerize into long filaments. So ZP2 and ZP3, they actually combine and polymerize into long filaments, and which are periodically linked by the cross bridges of ZP1. So periodically linked by the cross bridges of ZP1 like this. Now out of this ZP1, 2 and 3, ZP3 is very crucial, because so you will find they have an N-acetyl glucosamine residue. N-acetyl glucosamine residue on the surface. This is the binding site for the sperm, and sperm binds with this N-acetyl glucosamine residue of ZP3 with the help of another protein, that is galactosyl transferase, that is GALT1. GALT1 on the sperm and the N-acetyl glucosamine residue on the ZP3, they will combine with each other. Okay, so what happens after that? This GALT, that is galactosyl transferase 1, it activates the G protein that opens the calcium channels, and it initiates the acrosomal reaction. So acrosomal reaction means there will be exocytosis of the vesicular contents. Okay, and we will see that in this way that there will be fusion of this outer acrosomal membrane with the plasma membrane of the head of the sperm, and through this there will be exocytosis of the vesicular content like this. And what will happen after this exocytosis? You will find these inner acrosomal membrane. So acrosomal vesicle has got two membrane, outer membrane and inner membrane. Outer membrane was fused with the plasma membrane and helps in the release of the content. Okay, now after that what will happen, this inner acrosomal membrane, it will be exposed and it will act as the functional outer boundary of the sperm. So after acrosomal reaction, this inner acrosomal membrane will act as the functional outer boundary of the sperm head. Another important thing happens after this acrosomal reaction. So we can find a short actin filaments here. There are some short actin filaments. You can see here. Okay, short actin filaments. Now what happens after the acrosomal reaction? These actin filaments, you can see they are polymerized, and they are polymerized in this way, and they will form a long polymerized chain which is known as acrosomal process. So after acrosomal reaction, you will find in at the anterior aspect of the sperm, at the head of the sperm, there is formation of one acrosomal process which is surrounded by the inner acrosomal membrane, which is acting now as the outer boundary of the sperm head. Okay, so this is my acrosomal process. Now this acrosomal process and the outer boundary, they will penetrate this whole jelly layer of zona pellucida, okay, with the help of the enzymes released by this acrosome, and it is also facilitated by the vigorous mechanical propulsion by swimming movement of sperm's tail. Okay, and ultimately they will reach at the near the plasma membrane of the ovum. Now it is a time to fuse with the plasma membrane of the ovum. So what is the molecular basis of this fusion? If we look into the outer surface of this acrosomal process, you will find that there is a specific protein. Here, this is the binding protein. Okay. When this acrosomal process will try to fuse with the, first it will try to fuse with the outer vitelline envelope, vitelline membrane, and you can find that there are vitelline receptors. At this there are binding receptors on this vitelline envelope. So this binding protein, they will first form a bond between the binding receptors at the vitelline envelope. Okay, what will happen after that? After that, you will see this binding of acrosomal, this binding with the binding receptor will enhance the calcium activity released within the ovum cytoplasm. These are the cortical granules within the ovum cytoplasm. Now these cortical granules, now they will come on the plasma membrane, they will fuse with it. Okay, they will be fused with the plasma membrane. Okay, now due to the after the fusion, the content of this cortical granules, it will be released into this perivitelline space. You can see there are certain bonds in this perivitelline space. All these hydrolyzing enzymes, they will cause the destruction of this bond, and it will help the further penetration of the acrosomal process, and it will help the so that it will come in touch with the plasma membrane of the ovum. So what is so when the acrosomal process, it binds with the binding receptor of the vitelline membrane, what happens? Intracellular calcium concentration is increased, enzyme activity will be increased, so the cortical granules, they will fuse with the plasma membrane, they will release their content into the perivitelline space, and these content will cause a destruction of the bond, and it will help further penetration of the sperm, and sperm will ultimately come in contact. So sperm will ultimately come in contact with the plasma membrane. Okay, so these are the cortical granules which are released here, which are released into the perivitelline space. Okay, now after that, the sperm nucleus, it will enter within the ovum. Okay, but the process is not end here. It will not end here, because when one sperm nucleus has entered into the ovum, the next step is to prevent entry of other sperm into the ovum, that is to prevent the polyspermia. Okay, so how we can achieve this? So the block of polyspermia will be at two levels. One is the fast block due to the electrical change. Another will be the slow block due to modification of the zona pellucida. This will be by some chemical reactions. This we have already came to know, because these cortical granules, they will enter into the perivitelline space and they will release their content, and what will happen, that we will see later. But first come to the fast block. So why there will be fast block? When the sperm membrane and egg membrane, they will fuse, there will be influx of sodium. We know that positively charged sodium, if it enters within the cell, so there will be increased membrane potential will become positive, and this positive membrane potential will prevent the entry of sperm. So initial membrane potential resting was minus 75. After sodium entry, the potential will be plus 20 millivolt. So that potential will be denoted as the fertilization potential. So this intracellular positivity within the cell, this intracellular positivity will prevent the entry of the sperm. This is the first block. Followed by that, we have discussed that these cortical granules, they will exocytose in the perivitelline space. They will exocytose into the perivitelline space. So here these cortical granules, they have broken the bonds. Now the content that is released here, they will absorb water and they will swell up. So this will form an impermeable fertilization envelope surrounding this ovum, the fertilized ovum. So the content of the cortical granule, which will absorb water and it will swell up, and the whole thing will form. So this is your fertilization envelope. So first thing is intracellular positivity, that will prevent the entry of the sperm. Second is this fertilization envelope, it will also prevent the entry. In this way, in these two steps, the polyspermy is prevented. So now we know the process of fertilization. So first is the destruction of corona radiata, then penetration of zona pellucida via acrosomal reaction, then reaction between the binding protein and the binding receptor at the perivitelline, at the vitelline membrane. Then what will happen? The diffusion of the sperm nucleus, sperm membrane and the ovum membrane, and entry of sperm nucleus, and everything is done. We know after fertilization the meiotic arrest that was in the secondary oocyte, it will be withdrawn. So there will be release of ovum containing 23X, and there will be combination and formation of zygote. So combination of zygote. By 1.5 day, there will be formation of two cell stage. By 3 days, there will be 16 cell morula. By 4 days, 58 cell. Now they will be called as blastocyst. By 4.5 day, 104 cell blastocysts. Okay, these are the structure of the blastocyst. We can see the zona pellucida and the trophoblastic cells and the blastocele, the cavity, and the inner cell mass from where the actual embryo will take origin. And you will find at around 6th day they will be implanted, they will be embedded on the wall of the uterus. Okay, so now we are interested about this process of embed entry or invasion of this blastocyst into the wall of the uterus and how it is growing to form an embryo. So this is how the blastocyst is entering into the wall of the uterus. You can see the layers of the blastocyst. The outside layer is the zona pellucida layer. This outside layer is your zona pellucida layer, and inside this there is another cell layer, that is the trophoblastic layer. This is your trophoblastic layer, and this layer is your inner cell mass from where actually the embryo will take origin. So how this process is going to happen? So we will see that this developing embryo, once it is in contact with the endometrium, the blastocyst is surrounded by the trophoblast. Now this trophoblast is further divided into two layers. One is the cytotrophoblast, inner one, and the syncytiotrophoblast, that is the outer one. The syncytiotrophoblast is multinucleated mass with no discernible cell boundary, whereas the cytotrophoblast, it is made up of individual cells. Now this syncytiotrophoblast, you can see that this is the syncytiotrophoblast. It erodes the endometrium. So this is the endometrial stoma. It is the wall of the uterus. This syncytiotrophoblast, it erodes the endometrium and the blastocyst burrows into it. So this is the process of implantation. And the implantation site, it is usually on the dorsal wall of the uterus. So we can see from this picture. So this is the syncytiotrophoblast, this is the cytotrophoblast, and this is the inner cell mass, which is our future embryo. Okay, and it is the lumen of the uterus. Now at the beginning of the 3rd week, this syncytiotrophoblast, it generates an irregular finger-like projections. So we will find it. So this syncytiotrophoblast, they will generate this irregular finger-like projections which are lined internally by the cytotrophoblast. So internally it is lined by the cytotrophoblast and these finger-like projections are there. Okay, and these finger-like projections, finger-like buds, they are called the primary stem villi. And this primary stem villi, it will be surrounded by some lacunae. Okay, this lacunae which will form later the intervillous space where actually the maternal and fetal circulation, they will come and contact with each other. Okay, and there will be increased vascularity. So gradually the vascularity will be will develop, and uh, totally vascularized this villi. So we can see the vascularity here, where the connection between the maternal blood and the fetal blood. Fetal blood is coming through this umbilical cord, umbilical artery and vein. So this type of villi will be known as tertiary villi. This is totally fully vascularized villi. And this syncytiotrophoblasts and cytotrophoblast, these trophoblasts, these area will actually form the placenta and the fetal membrane. So they will anchor the fetus with the wall of the maternal uterus. So syncytiotrophoblasts and these trophoblastic layers, they will actually form the placenta and the fetal membrane. Now, the fetus and the mother, they are two genetically distinct individuals. We know that any kind of foreign body inside the body, our body immune system tries to reject it. That's why the graft rejection, reject any kind of renal transplantation or heart transplantation, we have to take so many precautions. We have to immunosuppress the patient so that the graft rejection should not take place. But what is happening in case of mother and the fetus? Fetus and mother are two genetically distinct individuals. But this transplant is very well tolerated in the body. The way the fetal graft is protected, this is very interesting. One important point is that is at the interface. One explanation is that at the placental trophoblast. So placental trophoblast is the interface between the fetus and the mother. So it separates the mother and the fetal tissue, maternal and the fetal tissue. It does not express MHC1 or MHC2. It does not express this MHC1, and we know that the HLA normal expression of this major histocompatibility complex MHC. So all cells, they will express this class 1. So when this is not expressing class 1, it will be identified by the as a foreign thing. So many immune system, that is a humoral immunity and the cytotoxic immunity, will try to destroy the cell. We know the normal immunity mechanism. So here at the trophoblastic interface, there will be expression of not this MHC class 1 and class 2, because these are very polymorphic and they can induce huge immune reactions. Instead of that, what is expressed? You will see that this interface will express HLA-G. So this HLA-G, it is non-polymorphic gene. Okay. So this non-polyformic HLA-G, if you see its function, it will inhibit the antigen presenting cells or APC. It will inhibit the NK cells, the T cells, all type of immune reactions it will inhibit. Therefore, there will be no development of antibodies against this fetal protein. This is very unique. Another explanation could be there is fast ligand on the surface of the placenta, this fast ligand. So when this fast ligand, it binds with the T cell, it causes the death of the T cell via apoptosis. So in this way, the immunity will be suppressed and fetal graft rejection will be prevented. Instead of rejection, mother will grow the fetus, will nurture the fetus, and it will form the mature fetus at term. Now, how the growth of the fetus will be facilitated? We know that normally in case of non-fertilization, the corpus luteum will start regressing from day 24 of a menstrual cycle. But if fertilization happens, this corpus luteum will form the corpus luteum of pregnancy. It will not undergo regression. Why this corpus luteum is necessary? Because it is the storehouse of the progesterone. So this progesterone is very much needed to protect this fetus. In addition to progesterone, it will release the estrogen, the relaxin, all of them, they will help to maintain the pregnancy. Now, who is keeping this corpus luteum alive? This is the very important hormone, that is human chorionic gonadotropin, HCG, which is released from the placenta. Okay, so how long this corpus luteum will do its function? It is seen that at around 6th week onward, corpus luteum will hand over its charge to the placenta. After this, corpus luteum will take the major function of producing all these hormones. Okay. So now the function of placenta is very important then. So it is releasing the HCG, which is actually initial part. It is maintaining the pregnancy. So the major functions of placenta is the invasion. Okay, invasion of the endometrium. It will give the nutrition to the fetus, and it will cause the production of hormones to maintain the pregnancy, local cytokine release. But in this segment, we will more focus on the hormone production ability of the placenta. What are the hormones that are released from the placenta? So the most important hormone we already came to know, it is HCG or human chorionic gonadotropin, which is keeping this corpus luteum alive. Now the function of the corpus luteum begins to decline after 8th week of pregnancy, but it persists. Although its function is reducing, it persists throughout the pregnancy. Now HCG secretion decreases after an initial marked rise, but estrogen and progesterone secretion increase until just before parturition. We will see it in the graphical representation. So here we can see that we have divided the pregnancy into 3 phases. First 3 months, that is a final, the first trimester, that is up to 12 weeks. Then the second trimester up to 6 months, 24 weeks. And then the last trimester, or the third trimester, that is last 3 months. Okay. So we can see that in the first trimester, the major hormone that is secreted is the HCG, and this HCG secretion, it is coming from the placenta, and you can see the level of HCG, it is so high. Now what is the function of the corpus luteum, the ovary? It is secreting the estrogen and progesterone. Okay, this estrogen progesterone, it is gradually rising. So after 12 weeks, that is 3 months of pregnancy, what will happen? We will see that placenta has started secreting the estrogen and progesterone. And what about the HCG? HCG is secreted, but its amount is less. Okay, so the this is the HCG level is reducing. Okay, that is written here. HCG secretion decreases after an initial marked rise. Okay, and the corpus luteum is still alive. It is secreting the estrogen and progesterone, but in less amount. Major portion will be secreted by the placenta. So, and their level is gradually, it is increasing. Now, after 6 months, what will happen after 24 weeks in the third trimester? You can see there is no contribution from the corpus luteum. All hormones are secreted from the placenta. And what is the proportion of this hormone? Estrogen and progesterone, their level is high. But what about the HCG? HCG level is less. So HCG, after initial marked rise, it will be declined. Although it will persist, but the progesterone and estrogen secretion, it will increase and increase until just before parturition. Now, human chorionic gonadotropin, from which part of the placenta it will be synthesized? It will be synthesized from the syncytial trophoblast. And after secretion from it, it will enter into the maternal circulation. It will stimulate the corpus luteum of the ovary, and that will increase the secretion of progesterone. Progesterone will prevent the menstruation and support the pregnancy. So this is very much needed. Now if we see the HCG, the function of the HCG, it appears in the maternal plasma as early as 6th day after fertilization. So we know that 6th day is the time of implantation. So the HCG, it appears in the maternal placenta as early as 6th day after fertilization, and it rapidly reaches the peak at the 10 to 12 weeks. We can see first trimester. Then the level decreases to 75% about 25th week and maintained, but its level is decreased, but it is maintained till down. Now this is the structure of HCG. If you look into in the detail of the structure of the HCG, you will find that this HCG, human chorionic gonadotropin, it is a glycoprotein. So we have gone through so many glycoproteins. Those are released from the anterior pituitary. They may be the LH, luteinizing hormone. They may be the FSH. All these are the glycoprotein. TSH. There are a structural similarity between all these glycoprotein. Although this is a placental glycoprotein and these are the anterior pituitary glycoprotein, there are very crucial similarity. All of them, they have alpha and beta chain, and this alpha chain is similar in all of them, in LH, FSH and HCG. By alpha chain you cannot differentiate them. It is the beta chain by which you can differentiate different glycoproteins. Now it contains the galactose and hexosamine. Regarding the function of this HCG, it binds with the LH receptor. So it has a structural similarity with LH. So it will bind with the LH receptor and it will maintain the corpus luteum. So luteinizing and luteotropic. It stimulates the progesterone production from the corpus luteum. Okay, and it plays a critical role in placental steroidogenesis, not only placental as well as the adrenal steroidogenesis of the fetus. It helps in the sexual differentiation. We know the steps of sexual. In my previous video you have seen the different stages of sexual differentiation. HCG stimulates the Leydig cells of the male fetus to produce the testosterone. This testosterone is very much necessary for development of external genitalia when it will be converted to dihydrotestosterone, that we know. Now it will, another important function of HCG, it stimulates the vomiting center. We know that in the first trimester the mother will face very a symptom that is known as morning sickness, vomiting tendency. So that in the early pregnancy, this morning sickness, it is contributed by presence of HCG. And the alpha subunit of this glycoprotein, it is 18,000 and beta subunit is 28,000. Now presence of HCG. So HCG level will be raised from 6th day of implantation. So from the 6th day of fertilization, and this presence of HCG in the maternal urine, it is the basis of pregnancy test. We all know that so many test kits are available. Few drops of urine, if you give into this test kit, they will tell you whether the mother is pregnant or not. The presence of this HCG in the urine, it is the basis of pregnancy test. How? That we are going to see now. So this is a normal pregnancy kit, you can see. So here is a area where where the urine drop is given. There are two areas. One is the test line, another is the control line. When the mother is, when the female is not pregnant, only the control line will give the color. But if the pregnancy is positive, there will be two lines like this. So this is a positive pregnancy, this is a negative pregnancy. Okay. So in normal pregnancy, HCG can be detected in both urine and serum as early as 7 to 10 days after conception. Okay, so after 7 to 10 days, the mother can detect whether the pregnancy has happened or not. And after that, the HCG level will rise very rapidly and frequently it will exceed 100 milli international unit per ml by first missed menstruation period. What do we mean by that? We know that now after ovulation, that is at 14th day, there will be fertilization, and from the fertilization, okay, so after 14th day of this ovulation, there is a time for menstrual bleeding. So if the pregnancy happens, there will be no menstrual bleeding, period will be missed. So when the period, actually mother become aware about the pregnancy when the period is missed. So at that time already 14 days has gone. Okay, 10 to 14 days. So at that time if the mother performs this pregnancy test, at that time the HCG level is very high, 100 milli-international unit per ml. Even at the 7 to 10 days after fertilization, this will give positive. So after the first menstrual period, it will definitely give a positive result, and it will reach its peak value and at around 10 to 12 weeks into pregnancy. Now, how does this pregnancy test it work? So this pregnancy test, it actually works via the principle of immunochromatography. It acts via the principle of immunochromatography, a unique two-site immune assay on the membrane. So here is the site where the urine drop is given. Okay. Followed by, so this is the strip, you can see this is the strip, here is the test line, here is the control line. Okay. And if the mother, so here at this area, what it contains? It contains the mobile anti-HCG antibody. Here there is mobile anti-HCG antibodies, and they are attached with some enzyme. So this is your antibody, you can see, and this blue rounded area is the enzyme attached to it. Okay. So if the HCG is present here, say if HCG is present in the urine, this HCG will combine with the anti-HCG. So there will be a combination between HCG, anti-HCG and the enzyme, and this part is mobile. It will be moved laterally due to the capillary movement, and it will come into the test line. Okay. So here we are having the mobile anti-HCG attached with an enzyme. So if the maternal urine contains the HCG, so there will be combination formation between the HCG and the anti-HCG and the enzyme attached with it. Now it will undergo the lateral movement due to capillary action, and it will come in contact with the test line. And what happens at the test line? In the test line, there are some immobilized anti-HCG antibody. Here there is immobilized anti-HCG antibodies are there. Okay, so this one is your HCG, and here at the test line another anti-HCG antibody is there. So it will again bind with it, and as it is immobilized, it is totally pasted, so this complex will remain on this area. Okay. So initially the mobile thing was anti-HCG and the HCG combined and the enzyme combined here, and this thing will come to an area or test line where an immobilized anti-HCG is there. It will again bind with this component. Okay. So there will be a complex formation here, and this enzyme which is attached here, they will actually give the color on the test line. Now if there is no such things which are did not bound with the extra amount of the mobile antigen which are not bound to the HCG, they will go further laterally, they will come to the control area, where another type of antibody which is a mouse immunoglobulin G, it is coated or immobilized, they will form complex with it and they will give a reaction and will give the color. If the mother urine does not have any HCG, so every unbound this antigen and antibody, it will move towards the control line and they will generate a color here, but there will be no color generation at the level of test line. Okay. So this is the principle. So initially it will be immobilized, then the unreacted conjugate and the unbound complex form a pink line, and if any move further, if it moves further the membrane and is subsequently immobilized by the anti-mouse antibody coated on the membrane at the control region. This control region is very important. It serves as to validate the test result. So this is the principle. So here if the HCG is present in the urine, so it will communicate with the anti-HCG antibody and the enzyme coated it, and then it will be immobilized on the test line. If it is not bound, so it will come to the control line where it is coated with another antibody. Now the test must be repeated. If one is not satisfied, the other line is very faint, it is not coming properly, then you have to use a fresh new test cassette. Okay, a clear background in the internal negative background control, a white to light colored background in the test area that does not interfere reading the test result indicates that the test is working properly. Now there are so many urine strip test kits are available in the market. Some are very sensitive. Even if the urine contains only 10 milli international unit of per ml of HCG, they will be able to detect it. So the range usually the range is from 10 to 100 milli international unit per ml depending on the brand. Okay, so most home pregnancy tests are based on this lateral flow technology. Now, what is the rules for specimen collection? Specimen collection, at least 1 ml of urine. It should be collected in dry, clean container with no preservatives. Then while specimen can be collected at any time of the day, but the specimen of choice is the first morning sample, as it has the highest concentration of HCG and is likely to produce false negative result. If the test cannot be performed immediately, so urine sampling can be refrigerated. Okay, and the refrigerated sample, when it has to be tested, it should come to the room temperature before testing. Now another important hormone other than the HCG that is secreted from the placenta, it is the human chorionic somatomammotrophine, or it is known as the human placental lactogen, or we can call it the chorionic growth hormone prolactin. The syncytiotrophoblasts of the placenta, it also secretes a large amount of a protein hormone. This hormone is lactogenic hormone, so human placental lactogen, and has a small amount of growth stimulating activity. It is called this HPL hormone. Okay, now it is seen that the structure, it is similar with the growth hormone, and it is seen that a large quantity of this hormone will be secreted in the maternal blood. Okay, but a very little will reach to the fetus. And what is the function of this hormone? You can see that it will increase, although it will increase the insulin resistance, it will cause the insulin secretion, it will cause insulin resistance. So insulin activity, it will not be there. So we know what is the function of insulin. Insulin is an anabolic hormone. It helps in the protein synthesis, it helps in the lipid synthesis, it helps in the entry of glucose into the cell. So when all these functions are inhibited, so instead of lipid synthesis, there will be lipolysis. Instead of protein synthesis, there will be protein lysis. Blood glucose level will be increased. And these products of proteolysis and the blood glucose, they will actually enter into the fetus and the help in the growth of the fetus. That's why it is also known as the growth hormone. Okay, and the products of the lipolysis, it will act as the energy source of the mother. Now so it is also known as the maternal growth hormone of the pregnancy. Now it is seen that if there is lack of secretion of HPL, there may be intrauterine growth retardation of the fetus. Intrauterine growth retardation of the fetus. Now we can see from here, so these are the hormones secreted from the placenta. We can see HCG is this hormone, and this is the HPL prolactin. So you can see it is gradually increasing in amount. Okay, and it is said that the amount of HPL secreted, so it is proportional to the size of the placenta. So placental weight, it is proportional with the size of the placenta, and which is normally weighs about 1/6 as much as fetus. And low HCG level in the blood, it signifies placental insufficiency. So placental insufficiency, that means fetus is going to become the growth retarded. So HPL is very important. Now other hormones from the placenta, it is a relaxin hormone. Relaxin in the early part of the pregnancy, it causes uterine relaxation, so facilitate implantation, prevent expulsion of the fetus. But in late pregnancy, relaxation of symphysis pubis and pelvic ligament will happen, that will facilitate the delivery, the expulsion of the fetus. And progesterone can also be released. Progesterone, you know, it will maintain the myometrial quiescence and promotes the continuation of pregnancy. Estrogen increases slowly in third trimester and reaches peak at term. So this in a nutshell, HCG, the progesterone and estrogen and their function. Now come to a very important concept, that is a fetoplacental unit. The fetus and the placenta, they interact in the formation of the steroid hormone. So mother, fetus and the placenta, they will act as an unit for synthesizing the steroid hormone. So as we have seen the two-cell theory of steroidogenesis in theca and granulosa cells, similarly here there is an interaction between the fetus, mother and the placenta to secrete or synthesize steroid. How they are interacting? We can see that the cholesterol here. So the cholesterol will come to the placenta. Placenta synthesizes the pregnenolone, okay, and the progesterone from the cholesterol. Some of the progesterone, you can see some of this progesterone, it enters the fetal circulation and provide the substrate for the formation of cortisol and corticosterone in fetal adrenal gland. So in the fetal adrenal cortex, they will help in the formation of cortisol. But some of the pregnenolone enters into the fetus, and along with the pregnenolone that is released from the liver, they will act as a substrate to form the dihydroepiandrosterone sulfate and the 6 in these androgens, that is, and the 16-hydroxy dihydroepiandrosterone sulfate. So this DHEA and 16-hydroxy DHEA, they will be transported back to the placenta, and from the DHEA there will be formation of estradiol. From 16-hydroxy DHEA there will be formation of estriol. They will be released into maternal circulation. Now since fetal 16-hydroxy DHEA, it is the principal substrate for the estrogen. In the urine of the mother, the amount of estriol is very important. This amount of estriol excretion, it can be monitored as an index of the state of the fetus. Okay, so whether this fetus and the fetoplacental unit, it is performing okay or not, that indication can be seen by measuring the urine estriol level. If the fetoplacental unit is performing well, that means that the DHEA and 16-hydroxy DHEA are generating in good amount. So 16-hydroxy DHEA will form estriol, and this is a predominant one. So this estriol in the urine of the mother, they will actually give an indication whether the well-being, indication of the well-being of the fetus. So this is very important concept. Now the maternal changes in pregnancy. First change, as we all know, the total there will be weight gain of the mother. The total weight gain during the course of singleton pregnancy, not the twin. So singleton pregnancy for a healthy woman, it averages around 11 kg. Okay. So 1 kg out of that, 1 kg in the first trimester, first 3 months, and 5 kg each in the second and third trimester. Then what are the other changes of different systems? First come to the cardiovascular system. The cardiac output is increased up to 40% and it persists till term. Systole stroke volume, it increases about 30%, peaks at 24 weeks. Heart rate increases by 10 to 15% and maximum at term. And systolic blood pressure, it also increases. Diastolic blood pressure falls slowly, reaches a nadir at 20 weeks. Now regarding the respiratory system, so this is a spirogram, that is a pre-pregnancy spirogram. You can see that there is significant decrease in the expiratory reserve volume, and the residual volume. So we know that residual volume and expiratory reserve volume, they combine to form the functional residual capacity. So the functional residual capacity will be reduced by 20%. Okay, whereas there will be increase in the inspiratory reserve volume and the tidal volume. You can see there is rise in the tidal volume, there is rise in the inspiratory reserve volume. So inspiratory capacity will be increased, and most importantly the vital capacity will remain unchanged. So some part is increased, so the vital capacity will remain unchanged. We know the vital capacity means tidal volume, inspiratory reserve volume and expiratory reserve volume. So expiratory reserve volume, although it is less, but these two factors will be increased, so the vital capacity will remain unchanged. Now come to the blood. Very important in case increase in the plasma volume due to expansion of the extracellular fluid volume. So how much? 400 ml increase in 8 weeks and 1 liter increase by 16 weeks as a result of sodium and water retention. So the fluid volume, fluid content will be increased. What about the RBC mass, cellular content? There will be increase in the RBC mass due to increase in erythropoietin. But the thing is that the increase in RBC mass, it is slower. As a result, the volume increase will be much faster and there will be a kind of relative decrease in RBC count. Relative decrease in RBC count, and this is known as a physiological anemia of pregnancy. Another important thing, increase coagulability. So increase the pro-coagulant activity, the factor, and there will be factor 7, factor 8, factor 9, factor 10, their level will be increased, and high coagulability may cause many fatal postpartum problem in mother. So clinician must be aware of these changes, physiological changes. Now there will be, in case of kidney, there will be remarkable increase in GFR, and GI motility is decreased. Mother often faces a fullness of abdomen. The size of anterior lobe of pituitary is increased. That is very important. Anterior lobe of pituitary will become very vascular. Okay, and if the postpartum hemorrhage is there, it can give rise to a very fatal syndrome. Okay, so there will be increase in the prolactin level. Demand for calcium, it will also increase. So during the time of pregnancy, mother should be given calcium supplement because there is increased demand for the calcium during the pregnancy. So this is a Sheehan syndrome. We can see if the postpartum, that after delivery, if the bleeding is very high, so blood volume will be reduced, and as during this time the vascularity of the anterior pituitary is very high, so it gets huge amount of blood. So any decrease in blood volume will cause reduction of blood supply and they will cause ischemia, and this ischemia will affect this highly vascular anterior pituitary. It's so much that it may undergo necrosis, and we know that any kind of pituitary, anterior pituitary is very important. It secretes so many hormones, it maintains pituitary gonadal axis, pituitary thyroid axis, so everything will be disrupted and the mother will suffer from a syndrome that is known as Sheehan syndrome. Now the process of labor. So we know how fertilization happened, how it was implanted, how the implanted blastocyst has grown as a full fetus. and what are the hormones that helped in the growth. Now it is a time for parturition or labor. So during pregnancy we will see the hormones such as progesterone, relaxin and nitric oxide maintain the uterus in the quiescent state. Okay, so there is no contraction of the uterus. Now prior to labor, you will see the changes in the estrogen to progesterone ratio facilitate activation of the uterine muscle. Ripening of the cervix occurs under the influence of prostaglandin, relaxin and nitric acid. So at the later part of the pregnancy, gradually the hormonal system, it prepares the uterus, the maternal system, for labor or parturition. Now following activation of myometrial cell and cervical ripening of the uterus, the myometrium responds to oxytocin, so many uterotonins, prostaglandins like this. Systemic plasma level of hormones are not responsive to local changes that initiate the maintain labor. Initiation of labor probably involves both fetal and maternal influence. We want to know what actually happens at the later part of pregnancy that initiates labor. So it is seen that from 35 weeks of gestation, there will be increased secretion of placental CRH, corticotropin releasing hormone. This plays a very vital role. This placental CRH will increase the cortisol level and dihydroepiandrosterone sulfate level, okay, in the fetus. So in the fetus, this placental CRH will stimulate the generation of cortisol and dihydroepiandrosterone. This dihydroepiandrosterone, it will go to the placenta and there will be generation of estrogen. Now this estrogen plays a very important role. This increase in estrogen makes the uterus very excitable. How it makes the uterus very excitable? It increases the gap junctions. It increases the gap junctions in the myometrium. It causes the production of more and more prostaglandins, which in turn causes uterine contraction. And most importantly, what it does, it helps in the it actually increases the oxytocin receptors at the myometrium or the decidua. So oxytocin receptor, their number, it will be increased in the decidua. Decidua means a part of the endometrium that is covering where the fetus is implanted. Okay. So oxytocin, what is the significance of increase in oxytocin receptor? You'll see that in the early labor, the maternal plasma oxytocin level is normal. It is almost equal to the pre-labor level. It is around 25 picogram per ml. But as there is increase in oxytocin receptor, it causes uterus to respond to a normal oxytocin concentration. Okay. So the function of estrogen, so first there is a placental CRH. It will enhance the dihydroepiandrosterone and their formation of estrogen. It is very important because it will increase the excitability of uterus by increasing the gap junction, by increasing the oxytocin receptor, by secreting the prostaglandins like this. And what is the role of this cortisol just before labor? This cortisol hastens the maturation of the respiratory system of the fetus. These every points are very important. Now what happens in the once the labor it is started, so there will be the cervix will undergo dilatation. Okay. So uterine contraction, there will be uterine contraction. It will cause a dilatation of the cervix. In addition to that, the head of the fetus pushes against the cervix. All these factors will cause the dilatation of cervix. And now impulse from the cervix, it will go to the brain. And from the brain, it will release the oxytocin from the posterior pituitary. this oxytocin is very important. What is the mechanism of action? So oxytocin has got its receptor on the myometrium. So it will bind with its receptor and intracellular calcium concentration will be very high. And you will see that this will result in a positive feedback loop. Positive feedback loop is established that aids the delivery and terminates on expulsion of the product of conception. So how this loop will act, you can say that uterine contraction, it will give rise to dilatation of cervix. Stimuli from cervix, it will increase the oxytocin. Oxytocin will again increase the uterine contraction. More uterine contraction means more oxytocin. So there is a very positive feedback loop formation. Okay. Now what is the function of oxytocin? It will enhance the intracellular calcium concentration, and by this it will stimulate the myoepithelial cell to increase the contractility. It will also enhance the local prostaglandin, that will also increase the myoepithelial, that will also increase the uterus to cause the contractility. In addition to that, the another function of oxytocin, and most very importantly, it will cause the contraction of the myoepithelial cell of the breast and cause the milk ejection, because once the baby is delivered, milk is the only food that is required for the baby. Okay. So in addition to the contractility, increase in contractility of the uterus, it will enhance the milk ejection reflex. So how it will do this, that we will read in the next section of lactation. Thank you.